name's Jenny Chow. I'm an environmental engineer major at Allorecognition College.
Hi, I'm Christina Soto Valderas, and I'm a math major at Santa Barbara City College.
Hi, I'm Kelly Tran, and I'm a biochemistry major at City College of San Francisco.
And I'm Ashley Wilburn, and I'm a computer engineering major at Allen Hancock College. And our project this week was dissecting allorecognition and Bacillus cerebrosei. Allorecognition is the ability to distinguish self from non-self in all organisms in the same species. To be more specific, it's the immune system's ability to be able to reject or accept a tissue as its own.
So many of you have probably heard about organ transplant, and you also probably know that it can be really difficult to find a good match between the donor and patient. Sometimes even if there is sort of a match, there's no real guarantee that the body will decide to function with the new organ. In the case of pregnancy, as far as self versus non-self, the fetus really only has half of the DNA of the mother. So why does the mother's body not reject the fetus? To better understand these cases in allorecognition, we're going to be studying the organism Bacillus cerebrosei.
In the lab, we are trying to understand allorecognition in Bacillus. As you can see the flower-like structure on the side, and the ampullae is the finger-like positions where allorecognition occurs. But Bacillus belongs to the family of Cooke, and it is the closest living relative of the vertebrate, as you can see on the tree of life, which describes the relationships among organisms. Understanding allorecognition in Bacillus is very important in helping us answer questions like how immunity evolves and develops, and understand more about how immunity works in humans.
Because of their simplicity, Bacillus is a great organism to study allorecognition in. Depending on their compatibility, they have the potential to either fuse at the ampullae, forming one whole system, or they will begin to develop dark scar tissue, ultimately rejecting each other. The lab has already identified several proteins involved in this process, but this week we will be studying the Bacillus histocompatibility factor, also known as VHF.
Okay, so what is VHF? Here is its amino acid sequence represented by single letter symbols. We took this sequence and inputted it into the XIT website that has a lot of tools that can help predict protein properties. So what we found is that there are no signal peptides that can help direct the VHF protein from inside to outside of the cell. There are also no transmembrane domain. There's also no transmembrane domain predicted that would tell us that it is embedded in the cell membrane. And finally, there is no functional domain predicted, so we can't infer its functions. So in short, we don't know its function or its properties. Our goal this week was to try and understand allorecognition in Bacillus by using a cell culture to express the VHF protein and to help localize it. This is the first step in characterizing the VHF.
In the lab, we introduced VHF genes, which was physically attached to red fluorescent protein M-cherry into the cells, and then we fixed the cells to maintain its cellular structure and prevent any other non-specific reactions from happening. And then we attach antibodies to the cells as a secondary method to confirm VHF protein's localizations in the cells, and we use secondary antibodies that emit green light.
So for our control in our first experiment, we used a red fluorescent M-cherry protein. So you can see here, the nucleus is blue and the M-cherry protein is located in the cytoplasm. So here we have the M-cherry that is physically bonded with a VHF protein, and it's now localized in the cell membrane. So for our second experiment, we used this result and used that as a way to compare if there's any specific region in the VHF protein that specializes in location.
So we use the shorter segments of the full-length VHF protein because we want to see if there are specific pieces that contribute to how the full length decides where to go. So we have here our data that we got from our experiments. 4 samples that go from the longest segment to the shortest on the right. So our first 3 samples told us that the protein, VHF and cherry, were located in the cell membrane. However, the strange thing was that our shortest protein on the far right there was present in both cell membrane and cytoplasm. So to further reconfirm that what we saw was accurate, we added antibodies to our samples. The antibody by itself fluoresces green, and when we combine the images with the fluorescence of the red from M. cherry, the images combine to show yellow, which shows that the localization of antibodies and the protein VHF M. cherry are one and the same.
From our data, we can conclude that VHF is in the cell membrane. Yet when we look at our shortest strands, we can see that it is located in the cell membrane as well as the cytoplasm. So to further characterize this protein, we could conduct an experiment to determine whether it resides inside or outside of the cell. Another way we could characterize it is to run an experiment binding the M cherry to the other side of the VHF using the same experimental methods we did here to see if there's a specific segment dedicated to localization.
So the more we find out about the VHF protein, the better we can understand the molecular mechanisms behind allorecognition.
And we would like to give thanks to our mentor, Daryl Takeda, our faculty advisor, Anthony De Tomaso,
Department of Molecular Cellular Developmental Biology.
And of course, we'd also like to thank everyone from the Cooke Bridges faculty to the RAs and everyone that's helped us. Also, thank you to our sponsors, Jack Kent Cooke Foundation, CNSI, CSEP, and also thank you to all the family and friends that have come to help support us.